I did not know David Barry. And unfortunately, did not have any opportunity to work with him when he was active. I did look up his history, and the one thing I did discover is that he was born in Nashua, New Hampshire, about 12 miles from where I was born. So that's the most I could find that we had in common. I've been asked today to recount much of the history of my discoveries and the research that I've been involved in, to tell anecdotes when possible, and that's what I'm going to do. Ray mentioned the viruses that I'm credited with discovering, and it reminded me, I didn't plan to say this, but I'll say it because it's kind of funny, that the one virus that got away, the one virus that I didn't, I was working in the office one day, and my wife called me to say that she had just taken our daughter to the doctors, and she had roseola. And I'm thinking, roseola, what the hell is that? So I went and looked it up, and it was a viral disease of unknown etiology. And I had already discovered SIV. I was on the way of discovering the KSHV-related virus. I said, oh, bring her in. It's another new virus we can discover. And my wife blew up and said, no way, you're not experimenting on my daughter. And it was about 8 months later that the Gallo lab reported the discovery of HHV6, the cause of roseola in people. So that's the one that got away. Okay, so my story starts with this 1981 publication in the New England Journal of Medicine by Michael Gottlieb et al. Reporting on this new disease that was called GRIDS, gay-related immunodeficiency virus, before it was changed to AIDS. And I remember like it was, I was a young assistant professor at Harvard working at the Primate Center. I was working exclusively on herpes viruses at the time. And I remember the director of the New England Primate Center at the time, Ronald Hunt, walking around the halls to whoever would listen, waving a printout of this article saying, this looks like what our monkeys have. This looks like what our monkeys have. And indeed, after asking more questions, I slowly, over the course of the next 3 months, 6 months, decided to start spending part of my effort to see if we could define the virus, a virus or viruses that could be a cause of that in monkeys. So it was a few years later, in 1983, that the New England group, headed by Norm Letvin, published in PNAS the initial description of this acquired immunodeficiency syndrome in our colony of macaque monkeys. And indeed, it turned out to be the major cause of morbidity and mortality in our primate research center and in other primate research centers around the country. And our competitors, our friendly competitors at the time from UC Davis, similarly published a paper around the same time or slightly before in Lancet, describing in their Davis, California Primate Center, similarly an acquired immunodeficiency syndrome that had many of the characteristics of the human disease. My lab chased down CMV, chased down other virus isolates we were getting from these immunodeficient monkeys, and then we hit on a new type D retrovirus that did indeed turn out to be the major cause of morbidity and mortality, and we published that in Science in 1984, just a few months before Gallo's series of papers in Science detailing HIV as the cause of AIDS. I have often teased our friendly Davis competitors at the time how that they also published a type D virus, but we published it first. And I wrote a sort of front page for our monthly newsletter at the New England Primate Center. I wrote a little article, sort of teasing them about this, how we published it first. And Preston Marx's objection was, well, the Lancet is published in England, and Science is published in the United States, and with the time difference, the 9-hour time difference with Davis, California, that it was actually published at the same time. All in good fun. The Davis Primate Center continued to chase the type D retrovirus as the cause and mechanisms. We went a different route, and we ended up going a different route because Norval King, the pathologist at our Primate Center, who was expert at electron microscopy, had electron microscope images from lymphoid tissue of some of the monkeys with immunodeficiency syndrome, and he was able to, he knew the morphology of the different subclasses of retroviruses, and he had EM pictures that clearly were of a lentivirus and not a type D retrovirus. So budding from the cell membrane in the absence of preformed nucleoids, you can see one here, and mature particles with a cylindrical or rod-shaped nucleoid. So we went after trying to recover this virus with this morphology in monkeys independently of the type D virus. The lentiviruses at the time were rather a little studied, obscure discipline with only a few investigators studying the lentiviruses from sheep, goats, and horses. And we were encouraged along this route when this paper appeared in 1984, early 1984, on the human AIDS virus, which was called, by the French group anyway, lymphadenopathy virus, showing structural and antigenic relatedness with equine infectious anemia virus, the horse lentivirus. And we were eventually successful, and we published in Science in 1985, almost exactly 1 year after the series of Gallo publications in Science on what was initially called HTLV3, and we called this an HTLV3-like virus. And after HTLV3 was renamed HIV, then our virus became SIV. I was senior author of this paper, and Dr. Daniel played the key role in doing the cell culturing. We partnered with the French to do sequencing. Sequencing was not as easy then as it is today, and we obtained full-length sequences of some of our isolates, and lifelong friendships with Pierre Sonego, Marc Alazon, and others at the Pasteur Institute. The Keith Mansfield at the New England Primate Center expended considerable effort in trying to define, well, where did this virus come from? What is it? And it turned out that macaque monkeys, which are an Asian Old World primate, are not naturally infected with SIV. And he traced the infection in the New England colony to a shipment of monkeys from Davis, California, wouldn't you know, which was received in early 1970, shipment of 5 monkeys who went on to develop AIDS and or lymphoma. In Dr. Hunt and Dr. King, in attempts to see if they could transmit that lymphoma, took some of the lymphoma tissue and inoculated it into other macaques, including 251, which is the commonly used strain SIV MAC 251, came from this monkey on the serial passage, and then from that isolate we obtained from monkey 239, and the virus is called SIV MAC 239. The current feeling, it's not published anywhere, but the sense is that how the virus got into rhesus monkeys at the Davis, California Center is Carlton Gadusek attempting to transmit prion diseases and prion-like diseases, did inoculations of materials from sooty mangabe monkeys, which is the origin of this virus, an African Old World primate, into rhesus monkeys, and that was it's generally believed that is how the virus got into rhesus monkeys at the Davis Primate Center. One of the next most important steps in this story is the isolation of an infectious molecular clone of SIV that was not only replication competent in cell culture, but fully capable of inducing AIDS in rhesus monkeys. And not only was this the first infectious pathogenic clone of SIV and HIV, it was actually the first pathogenic clone to be defined for any lentivirus. As sort of a side story, at the time there was a lot of controversy going on, generated by Peter Duesberg and a few others, challenging the HIV origins of AIDS, the viral etiology of AIDS, and our ability to induce the same disease, AIDS, in rhesus monkey using clone DNA, virus derived from clone DNA of defined sequence, provided unambiguous evidence for the viral etiology of this disease. I've been a big advocate over the years for the use of clone viruses as much as possible in rhesus monkeys, and it's not because our original SIV 239 clone still remains the best clone of choice to use for such studies, but using clone virus of defined sequence has enormous advantages. So you produce it in 293 T cells, there's no replication, there's no variation, it's defined sequence, there's 10,279 nucleotides. You know what those nucleotides are. If you do a neutralization assay, it's not a mixed population. We'll have sensitive and resistant populations in an uncloned virus stock. You can change any nucleotide, any set of nucleotides, any gene, any way you want to study effects on cell tropism, pathogenic potential, neutralization sensitivity, etc. So I consider our discovery and description of this pathogenic clone to be really important, and as much as possible, I encourage people to try to use clone viruses of defined sequence for their work. One of the applications that I'll talk about here that my lab has contributed to is using the clone virus to study the relative importance and functional role of the so-called auxiliary genes, genes that can be deleted from the virus and virus can still replicate. And we've studied a number of genes and genetic elements in this regard, and I put some of those publications on here. We've deleted Neff gene individually, VPR individually, VPX individually. We've made combinations. We've looked at the upstream sequences in U3 by extensive point mutations without changing the Neff coding sequence, as well as the transcriptional control elements, and have been able to define through those series of publications the relative importance and in some cases functional contribution of that gene or that genetic element. So after we had published the papers showing the contribution, the relative importance of Neff and the attenuating effects of deleting Neff from the virus, so virus with Neff gone is still not only replication competent, but it results in a persistent infection in rhesus monkeys, attenuated, lower undetectable viral loads, but nonetheless still a persistent infection. And around this time, Zhenjian Du came to my lab, and there was a strain that was being used by some people, an SIV strain called PBJ14, that was very unusual in inducing enormous lymphoid cell activation and an acute death. Animals died 14 days from this SIV infection. And Zhenjian came to my office and pointed out that there were 2 amino acid changes in Neff relative to SIV 239 that created a motif, YXXL, 7 amino acids YXXL, which is an SH2 binding domain, a domain recognized by Sark family proteins in signaling. And he proposed to change the 2 amino acids in 239 to see if he could reproduce that acute lymphoid activating properties. And in my great wisdom, I told him, you really think changing 2 amino acids is going to change such a dramatic phenotype? And I said, well, if you want to waste your time doing that experiment, go ahead. And fortunately, he didn't pay any attention to me. He went to the lab and did the experiment. And it turned out just changing those 2 amino acids reproduced the phenotype. Enormous lymphoid cell activation and acute death in 14 days in rhesus monkeys. So this prompted us to study, to look at a natural role, a role for natural Neff alleles, both SIV and HIV, in causing lymphoid activation. And in fact, we published this paper in Journal of Virology showing that a functional role of Neff is to increase, as an early gene product expressed early in the life cycle, is to increase the state of lymphoid cell activation, including the induction of IL-2 synthesis to create a more activated state of the cell to facilitate the ability of virus to produce its products and to replicate. So over this period of time, I met with my friend, Mario will recognize this pub outside of Worcester, Mass. I would meet with my friend John Sullivan once a week or so, one Wednesday night at his Irish pub, O'Connor's, and the next Wednesday at my Irish pub, Kennedy's. And we would talk science, we would talk politics, we would talk personalities. And I was telling him over our 4th or 5th beer about our monkey studies where we knocked out the Neff gene and it's still infectious, they're still persistently infected, but it's highly attenuated and basically elite controllers. And Sully was in charge of the hemophiliac cohort in central and western Massachusetts. And he said, you know, I have a few people in my cohort that look like they're elite controllers like that. You want to look at them. And on the 7th or 8th beer, I agreed, okay, let's provide 5 samples and we'll take a look at it. And Frankkershoff was the person in my lab who signed on to work on that, took the 5 samples, and he came to my office to show me the PCR products from the 5 patients. He did several genes, but including the Neff gene. And he said, look, one of them has a big gaping deletion in Neff. And my reaction, of course, was another moment of brilliance where I said, oh man, you contaminated it with our Neff deleted HIV-1 clone. Get some more samples and go back and do it again. But it turned out all this guy ever had was Neff deleted, was HIV with a deletion in its Neff gene. John had samples from this patient dating back to his first visit in 1983, and the primordial deletion, the primary deletion in this individual was, I think it turns out to be 122 or 124 nucleotides from the Neff unique region, that is the portion of Neff between envelope and the start of the LTR. And by looking at sequential samples over the course of time, Frank was able to show that the virus progressively lost the rest of the Neff coding sequences that it no longer needed. So the virus in that individual retained the polypurine tract, retained the U3 terminal sequences, retained the NF-kappa B SP1 binding sites, but basically spit out the rest of these upstream U3 sequences in the LTR. So this provided evidence, reasonably good evidence, that really the only functional role of these upstream U3 sequences is to serve as Neff coding sequence. And we followed this experiment up by, in SIV, extensively mutating the U3 in a way with synonymous codons that retain the Neff coding sequence, and that heavily mutated U3 region still behaved totally like wild-type virus, consistent with the role of these upstream U3 sequences being only Neff coding sequence. These sequential deletions over time in this HIV-1 person actually mimicked what we had published a few years earlier in SIV, showing that in Neff-deleted SIV, these upstream U3 sequences become progressively deleted over time, and that's shown here. Here's the deletion that we started with, and you can see over time that the virus spits out the rest of these sequences that it no longer needs. Now, most people don't remember the Sullivan-Frankkershoff, Sullivan-Desrosiers publication on this first human infected with Neff-deleted HIV, because the Australians came up with a much more even remarkable set of circumstances. They heard about our report, they saw our report, and they went looking at what is called the Sydney Blood Bank Cohort. This is a single blood donor who turned out to be HIV infected, who was responsible for infecting 8 people with HIV from his blood donations. Both the donor and the 8 recipients were all elite controllers, all long-term non-progressors, and it turned out they too, all 9 of them, had virus with a deletion in its Neff gene. And they, subsequent to our papers, also showed progressive loss of the rest of the Neff coding sequences over time. Given the attenuating effects of deleting Neff, deleting VPR, deleting combinations of genes, it was only natural for us to think of using this virus, at least in monkeys, in vaccine challenge experiments. And we did that, first published in 1992. Again, Dr. Daniel is first author and me as last author, showing the protective effects of live attenuated strains, including just the single Neff gene deletion in protecting against challenge by wild-type disease-causing SIV. To this day, live attenuated SIV remains the gold standard of vaccine protection in the rhesus monkey model. The gold standard that all vaccine approaches are trying to achieve. So it worked very well. Discussion started about whether this should be considered for human use. And I had the good fortune around this time to meet Albert Sabin, the discoverer of the polio vaccine, and started a communication with him. Basically started with my saying something like, Dr. Sabin, you of all people should appreciate the potential of a live attenuated virus as a vaccine. I've used some discretion here and not shown the letter he sent me where he called me a stupid shit, but he was pretty well known as being a crusty character. But I did show this one where he says to me in his handwritten letter, although you pay me the courtesy of writing to me, you pay no attention to what I say or publish. And he, and some of the publications he was referring to me were a little hard to find. Here's one published in 1954, which with great effort I was eventually able to locate. But I still have all of these letters in my folder at home. The World Health Organization convened a working group to consider the concept of live attenuated vaccines, and I show the conclusions here. I'll just read 4 and 5. 4, the development of live attenuated vaccines should be intensively explored in parallel with other vaccine approaches. And 5, additional preclinical data, preclinical meaning monkey and chimpanzee, should be obtained in the macaque and chimpanzee models using attenuated deleted variants of SIV and HIV. But perhaps those of you who are old enough to be around then know that this turned out to be a pretty controversial discussion, and the idea was squashed. And I think squashing of the idea for use in people was totally appropriate. but I think over the years there has been much to be learned from use of live attenuated strains as vaccine strains in monkey studies, and I kind of wish that NIH and the governing bodies had paid more attention to this World Health Organization in funding more studies to study live attenuated vaccines in the context of animal models. I will make 2 points regarding the concept of live attenuated vaccines for HIV. While SIV delta Neff and delta 3 is Neff deleted, VPR deleted, and upstream U3 sequences deleted, provide strong protection against challenge by SIV strains identical or closely matched in sequence, these live attenuated vaccines do not provide very good protection against heterologous SIV strains, like the ones one would expect to see in a natural sort of exposure setting. So as good as live attenuated is, and again, it's still the gold standard, it does not do very well in protecting against heterologous strains. And this situation is probably analogous to the inability of infection by one HIV strain to routinely protect against superinfection by a different HIV strain. And perhaps the best work along these lines in humans is the work of Julie Overbaugh, where she has over the years studied a cohort of 56 HIV-infected women in Mombasa, Kenya. And she's documented, I think the number now is up to 13. She's documented 10 at the time this old slide was made. I think it's now 13 cases of the 56 women where they became superinfected by different strain of a different sequence. And by studying them over time, you can see, okay, they've got one heterogeneous mix of sequence. This visit, the next visit, the next visit, bang, a new virus appears, different in sequence. And that was usually associated with a dramatic jump in viral load. Current uses of SIV and SHIV models include better understanding of pathogenesis. So if one has questions about role of macrophage tropism, if a gene product like the Neff gene product has 5 or 6 reported functional activities, one can separate those with defined mutants. A role of escape from neutralization, etc. can be studied in these models. They can be used, very importantly, to inform and guide development of vaccine concepts for use in people. I'm sure most of you know that there are lots of scientific barriers to make a vaccine that'll be successful against HIV, where even infection with one strain does not protect against superinfection with another strain. What are we going to put in a vaccine that'll make it work? And spending $150 million per efficacy trial in people is, in my opinion, not the best way to go. And I think we should be trying to get more out of the monkey models to inform and guide development of vaccine concepts for use in humans, where the most promising concepts can be forwarded to trials in people, and investigation of novel therapy strategies, including cure strategies. And that will be the subject of my talk tomorrow, and the use of viral vectors for delivering antibodies toward long-term viral suppression. That just says the same thing. And the vaccine concepts in my lab include recombinant herpes virus, and the concept there is herpes viruses are forever, that immune responses to them are up, on, and active for life. And if we're ever going to have a vaccine that elicits protective immune responses, I forgot to mention that Albert Sabin actually didn't believe that immune responses had anything to do with the protective effects of the polio vaccine. So that made my arguments a little bit difficult with him. But recombinant herpes virus at least has the potential of being up, on, and active. And then, and the subject of my talk tomorrow, use of AAV for long-term delivery of monoclonal antibodies. I would like to say that I'm enormously proud of the people that have trained with me over the years. They include Jay Jung, Frankkershoff, Blossom Damania, Welkin Johnson, David Evans, Michaela Gack, and Alexander Hahn, and without whom none of this would be possible. Thank you very much.